Welcome, everyone, and thanks for joining us today. My name is Heather Pinkett. I am president of the Protein Society, and I just wanted to take a few minutes before we dive in and share some exciting news about some upcoming events for the Protein Society. So there's a lot to look forward to. So first and foremost, we have our annual symposium. This year, it's our 40th anniversary, and we have a lot of great things planned for this milestone year. Just wanted to make note that abstract submission deadline is March the 23rd. We have space for open talks from these abstracts, and also we are offering travel awards. So make sure you apply by March 23rd. It's going to be a really great meeting in Boston this year at the Western Seaport. And the date is July 19th to the 22nd. Okay, second announcement. Mark your calendars for April 27th, Monday. We have another follow-up webinar. So this particular webinar is organized by Annalisa Pastor at King's College London, and it is going to be on chaperones. And this will be a really exciting webinar, so mark your calendars. Our third announcement, of course, is going to be just a word about protein science. Our society continues to have an outstanding resource for the protein science community with our journal, Protein Science. It is the premier journal for all things proteins. And, this is really led in part by John Currian and a wonderful team of our editorial board. And so I really want to just reach out and say, if you're thinking about submitting a paper, Protein Science would be a great journal to consider, especially if it fits into our areas of interest. And last but not least, of course, is our webinar today, which is our Protein Science Tools, which is a special issue that we put out every year in Protein Science. And today we have a wonderful array of topics to be covered. All came out of articles out of our premier Protein Science Tools issue. So I'm going to turn it over to our organizers today, who are going to give us a short introduction before we let each of our four speakers provide their talks.
I did want to also welcome you and make sure you're in the right place. As Heather mentioned, this is our tools issue. Protein Science Journal is incredibly important to the society and the community, so thank you for joining us. This is an overview of our tools issue, which is published yearly. Of course, it's computational tools, and you get a little bit of information about what we're trying to do. This is one of our most popular and cited issues, which started many years ago. So we're really glad that we can provide this for you. Here, the issue has grown, and NIR is already working on future issues. You can access the tools issue online from our website and from Wiley, who is the publisher of Protein Science. And we're very proud to have 54 papers in 2025. And it is ongoing for the most part by invitation, but you can reach out to the Protein Society or NIR directly. And we aim for 100. So that's compared to previous issues, that will be a huge success. And taking us to our webinar today, we have Dr. Shengyou Huang, Dr. Gajendra Raghava, Dr. Matteo Tiberti, and Dr. Luca Genz joining us. And as you can see their talk titles right here, but they will give you a lot more information about the work that they're doing. So it will be more interesting when they do it rather than myself. And last but not least, we do have a mini biography of our speakers as well as the speaker order. So I will just let you take a look and I'll scroll through.
Everyone, thank you for joining this webinar for Protein Science Special Tours. My name is Shengyou Huang from School of Physics at Huazhong University of Science and Technology. First, I'd like to thank the Protein Society for organizing this great webinar and also thank Nia for inviting. Today, I'm going to talk about model building of protein complexes from cryo-EM maps with one of our recently developed tool input. Before introducing our input method, I'd like to briefly introduce the research in my lab. My lab has been developing computational tools for modeling of biomolecular interactions. My research interests including molecular docking, complex structure prediction, and cryo-EM modeling. For each of them, we have developed different computational tools like HDoc, DeepInter, EMReady, and so on. Today, I'm going to talk about one of our cryo-EM modeling tools, input. First, I'd like to give some background about cryo-EM, which won the 2017 Nobel Prize in Chemistry. Here is a simple workflow of cryo-EM structure determination. First, we prepare the sample of proteins. Then we take photos of the protein sample using the cryo-EM equipment. After that, we got a lot of two-dimensional single-particle images of the protein. With the 2D images, we can reconstruct the three-dimensional density map of the protein using tools like Rely-On or CRISPR. Until now, we have the three-dimensional density map of the protein, but we still don't have the atomic model of the protein. Therefore, the last step of the cryo-EM is to build atomic models from the three-dimensional density map, namely model building. That's what we are doing here. However, building atomic models from cryo-EM maps is challenging because cryo-EM maps have three limitations. First, compared with X-ray crystal, cryo-EM maps normally have much lower resolutions. Second, cryo-EM maps have no single-to-noise ratio. Third, cryo-EM map is very heterogeneous. That is, in the same map, some regions have high resolutions and some regions have very low resolutions. Therefore, model building of cryo-EM is often a very challenging and labor-intensive task. So, according to the EMDB and the PDB databases, many cryo-EM maps don't have an atomic structure. The number of the cryo-EM maps, here the purple one, is about twice the number of cryo-EM structures. Here the green one. There is a big gap between maps and structures. Therefore, computational tools are very needed for building atomic models from cryo-EM maps. Our input is exactly one of such tools. Generally, cryo-EM maps have a very wide range of resolutions from 3 angstroms or better to as low as 20 angstroms. Therefore, different strategies have been developed for modeling building of different resolution maps. For high resolution, such as 3 angstrom or better maps, de novo modeling can be used to determine the atomic structure directly from the map. Its advantage is the build model is very accurate. However, as I mentioned before, cryo-EM map is very heterogeneous and therefore often includes many low-resolution regions, even though its global resolution is high. De novo modeling will fail in these low-resolution regions. For medium to low resolution maps, they normally don't have enough atomic details. Therefore, template-based modeling is often used. That is, a template structure is first extracted from the PDB or predicted using structure prediction tools like AlphaFold. The template structure is then fitted into the map, followed by post-refinement. The advantage of template-based modeling is it can build more complete structures in low-resolution regions. However, because proteins are flexible, so the fitted structures may not necessarily reflect the true density map. Therefore, we tried to combine the advantages of both de novo modeling and template-based modeling and proposed our import method. Here is the workflow of our import method. The upper part is the de novo modeling pipeline, specifically starting from an input query map. We used deep learning to extract the C-alpha, residue type, and mention probability maps from the raw map. Due to the time reason, I'm not going into the details of our deep learning network here. Then the C-alpha probability maps can convert it to the C-alpha positions using the mean shift algorithm. The all-item model can be generated from the C-alpha positions using the all-item prediction module. The lower part is the pipeline for template-based modeling, specifically with provided protein sequences. Template structures are predicted using tools like AlphaFold. Then the template structures are fitted into the mentioned probability maps and the backbone frame built by the de novo modeling part. So finally, the all-item model can be obtained after fragment assembly and post-refinement of fitted structures. This way, by integrating two strategies together, we hope to take both the accuracy advantage of de novo modeling and the completeness advantage of template-based modeling in our import method. In addition to the integrative modeling strategy, I'd also like to point out two special modules that distinguish our import method from others. One is the all-item prediction module here, which uses a 3-track attention network. The second one is the fragment-based structure fitting module. Here, I will introduce them in more detail in the following. This is the all-item prediction module. Basically, we use a 3-track attention network to efficiently integrate the density map, C-alpha position, and the backbone frames during all-item prediction. Here, the one-dimensional node feature is the point density of C-alpha positions. The 2D edge feature is the density between two C-alpha positions. The 3-dimensional feature is the backbone frame built by the de novo modeling method. Here is the 3-track attention network used in our import method, which integrates 1D, 2D, and 3D features. We borrowed it from the protein structure prediction method Rosetta Fold, developed by the Big Group. For more information about this network, users may refer to its original paper. This is the fragment-based structure fitting and assembly protocol. Here, we performed two types of structure fitting. One is to align predicted structures with de novo modeled structures. That's structure to structure alignment. Here, this part. The other is to fit predicted structures into the mentioned density map. That is the structure to map fitting. Here, this part. For both fitting, we did fragment-based fitting to consider protein flexibility. This way, we can recover the structures that are missing in de novo building models. Finally, the all-item model can be generated after fragment assembling and post-refinement of fitted structures. That's all about our input method. Now let's see the performance of our input method. To benchmark our input, we tested it on the benchmark of 175 cryo-EM maps from the model engine study and compared it with state-of-the-art approaches. The first evaluation is for structure recovery capability. Panel A is the backbone coverage regardless of residue types. Our input builds 91.2% of the structure, which is significantly higher than other methods. Panel B measures the sequence match, that is, whether or not the amino acid types are correctly identified in the recovered backbone. Again, input obtains the highest success rate of 95.87%. Panel C shows the sequence recall, that is the product of backbone coverage and sequence match, measuring the overall performance. In this regard, input also performs the best, giving the sequence recall of 87.79%. Here is one example of for EMD 15220. The green one is the PDB structure. The blue one is the build models by different methods. On this tact, the build models by our input overlaps very well with the PDB structure, giving an excellent sequence recall of 91.06%, while other methods show lower sequence recalls because of some failed regions as indicated by the dashed box. Here, and here. Next, we measured the accuracy of build models in terms of TM score and RMSD. For TM score, here panel A, the higher value is the better. Our input gives the highest TM score of 0.9 among all tested methods. For RMSD, here panel B, the lower value is the better. Again, our input shows the best RMSD of 1.42 angstroms. Here is one example for EMD 15690. Here, the gray one is the PDB structure. The colored ones are the build models. On this tact, our input achieves a high TM score of 0.888, while other methods show lower TM scores. Specifically for diplomat, the build model mismatched with the PDB structure for the red chain. Here, for Phoenix and AlphaFold 3, the models missed the up domains here and here. The third evaluation is to make sure the build models are reasonable in terms of structure geometry, such as atomic connections, bond lengths, and bond angles, and so on. We used the mole probability program to calculate the values of different geometry metrics. In this table, up arrow means the higher the better. Down arrow means the lower value the better. It can be seen from this table for de novo protocols. These two, whether or not refined, our input is comparable to model engine. However, for integrated modeling here, the full pipeline, our input performs better than the other methods. Another important evaluation is to make sure that the build models are consistent with the cryo-EM map. This is very important because we are building atomic models for the map. Therefore, the build model must be faithful with the map. Here we use the correlation between the build model and the cryo map, including CC mask, CC box, CC peak, and CC volume, to measure the accuracy of the build model. These correlations are calculated using the Phoenix Map Model CC program. Again, it can be seen from this table. For de novo protocols here and here, whether or not refined, our input is compatible to the model engine. However, for integrated modeling, the full pipeline here, our input performs better than the other method. This is one example for EMD 27661. Here, the green one is the PDB structure. The blue one is the build model by input. The yellow ones are the ligands, and the red mesh stands for the density. It can be seen from this figure. The build model by input not only overlaps very well with the PDB structures. Here, see the enlarged view, the two panels, but also fits the density map very well. Here, show the enlarged view. These results demonstrate the high accuracy of the build model by input. Moreover, our input method can also give a confidence score for each residue of the build model, so that users can easily do quality control. Here is the build model for EMD 33854, colored by confidence score. Generally, the high confidence score is the better. However, it should be emphasized that the low confidence parts do not necessarily mean the non-arc structures, but often indicate low resolution regions in the map. Therefore, users may need to double check the build model in these regions. So far, I have introduced our input method. Now, where to get our tool? Users can directly download our input program directly from this GitHub website. It's totally free for both commercial and non-commercial users. It requires a GPU to run. The installation is very simple and straightforward. If you don't want to install, you may also run a quick trial on the Google Colab site. So we have a demo, so you can click here to get into the demo. Our input is also very user-friendly. So just one command for all. After installation, users may type input build --help for help information anytime. The minimal required input is a QuiEM map. The sequences and the structures are optional, but more input is better, so it's more accurate for the build model. There are three ways to run input. Those are modeling with sequences, and the modeling without tagged sequences, and the modeling with tagged sequence and predict structures. So the third one is more accurate. So here are three examples to show how to run input. So user can directly download the files and try them on their own. Okay, I think that's all I'd like to share today. This input study was mainly done by my students Matteo, Chenji, Hao, and Huang. In addition, my former student Jiahua laid a good foundation for our cryo project. I'd like to thank them all for the hard working on this study, and also thank the funders, National Science Foundation of China, and our university for supporting this work. Okay, finally, thank you all for your attention, and I welcome any questions.
Shiyu, thanks for that talk. That was absolutely wonderful. So I'm
Thank
going to
you.
start off with the questions that are in the
Okay.
Q&A section, but also remember to state that if you do have a question, make sure you put it in the Q&A. So
Okay.
the first question is very impressive. How well does EM-PROT perform on non-protein macromolecules in the density maps?
So non-protein so means the like the nucleic acids or ligands. Yeah, I guess so. And right now, we import can model the protein only, but we have other tools like mRNA for nucleic acid modeling. So for ligands, we still don't have a tool for that yet, but it's an ongoing project for us to do that. I guess right now we have united tool for both proteins and nucleic acids. Uh, modeling. But probably it may be released after 1 or 2 months. But for ligands modeling, we are still doing that. So
Excellent. Thanks. Just a follow-up question there. When you're thinking about ligands, what is some of the challenges that you anticipate with being able to build in a ligand that using the de novo building approach may be able to solve?
well, that's a very good question. So very good question. So actually, because we are so using deep learning method to for our tools. So and the training data is very important for ligands. One great challenge is so uh, the chemical space of ligands is very large because for proteins we just only have 20 amino acids, so that's limited. But for ligands, so there is a huge number of ligands. So there are so many types. So it's hard for the deep learning to find a pattern. So for the ligands, but even though it's difficult, but we are still trying to develop a method by combining the physics and deep learning. So, for example, combine docking. So right now we have a kind of design. So, and first we identify the ligand density, and if user can let us know what ligand that is, we can model the ligand into that density. So we can do that right now. But the de novo one is very challenging.
Yeah, great.
Okay, thank you.
Thanks. We have two other questions that I will ask quickly. So, how do you assess the accuracy of EM density maps? It sometimes contains low density, such as in a bit flexible regions. How do you decide when to trust computer predictions or the EM density?
Uh, this is a good question. So actually, right now for us, um, we tend to trust the density more than the model actually. So after we build the model, we will assess the accuracy by comparing between the model and the density. If it fits well, so we think it's good. If it does not fit the density well, so we just give it a low confidence score and alert users to double check that.
Great.
So basically we tend to trust the density. So
Always trust.
probably somebody may think the density is not accurate either. right? So that's actually, so we can right now we can only trust the density.
Yeah, great. Thanks. And the last question is, what is the lowest resolution that EM Pro will still perform well?
So on the I guess probably if the as long as the notion is functional or better, it will perform well. So and but I think it can work for the maps with resolution as low as 8 antrams. So and but the accuracy will become worse. So another way to rescue the accuracy is so if users can provide the predicted structures by AlphaFold 3, like that, something like that. So the accuracy is still good. So like it can get a 90%. So as long as users can provide the predicted structures. So for de novo one, we recommend the maps with resolutions 4 antrams or better.
Okay, great. Thanks so much.
Okay, thank you.
I'll save the last question until the end of our webinar and let our next speaker share their screen.
Okay, thank you.
Thanks so much.
My screen visible?
Yes, it is. Thank
Okay.
you.
Okay. So what I should say, it's a good night for me, good morning for the others. So it's a different time zone. Okay. So here is the talk. We are talking about decoding of proteins. So I will talk about the 3 points: structural, function, and therapeutic annotation. So I will go one by one. Structure, function, and therapeutic annotation means their applications. And our group mainly involved in 3 type of activities: development of software, development of biological databases, and web servers. So this is our website. If you are interested in our software server, you can use them. These slides were created using the various resources. So we are not claiming any authorship on any slide. Okay, you can use it because I believe in open source software. Regarding the Protein Science Journal, I am associated with this journal from the long time. Let's say we have the first paper in this journal in 1997, long time back. Then we have the method in 2003, a neural network based method for predicting the gamma turn actually. And then it was a tool or the beta turn prediction. Though they have started the tool issue recently, but we are already publishing our web-based tools in this journal actually. So we are associated with this journal from the long time. Not that we are not associated now. We are also associated at this moment also. So last year itself, we published the paper on the predicting a neurotoxic peptides as well as the antibody human specific B cell epitopes. Okay. There is a other methods like NAG binder. So whole aim is the Protein Science is excellent journal. It review very well. Whatever the paper we have published in the journal, they are very well taken by the community and they are heavily used by the community. So I will encourage you to publish more and more paper in the Protein Science. Excellent journal. Okay. Now I am coming on the peptide and protein. As you know, peptide is nothing. If it is smaller than 50 amino acid, we call peptide. If it is more, then we call proteins. But they have the application everywhere. As it is known that protein and peptides are called the living organism, means servant of living organism, means they are there in every part of organism basically. You cannot think about the biological activities without the proteins or peptides. Okay. So now, first I will cover that our group is involved in the protein structure prediction. We participate in the most of the CASP competitions over the years, and we have developed the different type of tools that like predicting the alpha terms in the proteins, secondary structure in the proteins, beta terms, many type of prediction methods we have developed over the years. Okay, so we are there in the protein structure prediction over the time. So you know very well that after this AlphaFold 2, where they demonstrated it worked better than any other method in term of the protein structure prediction, then most of the traditional groups like our group, we have shifted our focus towards some other areas rather than focusing on the protein structure prediction. Okay. So one of the activity our group is involved over the years, instead of competing with the protein structure prediction methods over the years, we have focused ourselves on the peptide structure prediction, means when its protein is small, because they have the different type of conformations. Not only that, because if you talk about the protein or peptide based therapeutics, then question comes, what is the stability of the protein and peptide into the body fluids, basically. That's a major challenge. And how you can increase their half-life or stability in the body fluid depend upon their modifications. So natural peptide or protein have the limited half-life. So we have to do some chemical modifications, like we can add the D-amino acids, we can add the terminal modifications like N-terminal modifications, C-terminal modifications, we can do the cyclizations, we can add the non-natural residues, PTM, blah, blah, blah. But the question is, once you add these modifications, what will be the structure of the peptide? That's a challenge. So this tool is the only tool which allows wide range of modifications in the peptide and predict its structure. So it is heavily used by the community, and we are coming up with the new version of this tool, and where we demonstrate that its performance is as good as any other methods. Okay. The next question comes. Protein structure prediction is one part where you are talking about the structure of the protein. Maybe it's secondary structure, beta term, or maybe tertiary structure. But there is another interesting part, protein interaction, because protein interact with a wide range of molecules. So here we have developed the number of methods for predicting the protein residues which interact with the different type of molecules. Let's say, for example, ATP predict the ATP interacting residues in a protein. Identify the ATP binding sites in ATP binding proteins. Similarly, glyco-EP predicts the glycosylation sites in eukaryotic proteins. For glyco-PP is another example of prediction of glycosylation in prokaryotic protein. Same way, we have the methods for predicting a DNA binding protein, RNA binding proteins, wide range of methods are there actually. So we have developed the method where we are predicting interaction of the different type of molecule with the protein. And all these tools are freely available to the community, and it's an excellent web server is there, user-friendly web server is there, so anyone can use them. Same way, there is another thing, is function. That what is the function of the protein? Structure is fine, because most of the time we predict the structure of a protein so that we can understand the function. But there is a possibility that you can predict the structure of the function of a protein directly. So here is some examples. For example, this tool, NRPRED, allow you to predict and classify nuclear receptors. Okay. Same way, GPSRPRED allow you to predict the family and subfamilies of G protein coupled receptors. These are the specific type of proteins. ESL-PRED allow you to the subcellular localization of eukaryotic protein. So we have the wide range of softwares for the community so that you can use them for day-to-day work. Now, I am coming on the another interesting topic, because I believe structure and function is more and more annotation-oriented work, where you want to understand the function and structure of the protein. There is another side of the protein and peptide. These protein and peptides can be used as a therapeutics. Because why I should to go for the small molecule-based drugs? I can go to the protein and peptide-based drugs because I understand protein better than the small molecules or small chemicals. So what we did long time back, we created a database of FDA-approved therapeutic peptide and protein, and we provide the comprehensive information. So the community is heavily using this database for understanding the therapeutic peptide and proteins. If you see carefully, the application of peptide and protein can be divided in 4 categories. Category number 1, when you have the particular protein, but it is defective, or your system is not making sufficient protein. Good example is insulin. In diabetic patient, insulin is not sufficient made by the person. So we provide the insulin from the outside. So we are providing the aid from the outside. So this is one major application. Another is peptides as a self-contained peptide are used for the drug delivery, because these peptides have the capability that they can take the molecule into the within the cell actually. Okay, so that's another application. Third application everybody know very well is vaccines, because protein and peptide based vaccines are heavily used. And fourth is diagnostics. So it means that they are protein and peptide are widely used for the therapeutic purpose. Here, I will give you the example of vaccine, which is my favorite field. Over the last 25 years, I am working in this area and still working in this area. Okay, so I can give you the good example of vaccines. Recently, we have the COVID-19. At that time, vaccines have been developed. In India, our country developed the vaccine called Covaxin. This Covaxin was based on the traditional culture, where we have taken the full virus, we killed it, we annotate it, and we give as a vaccine candidate. That is called traditional technique, where whole organism is used as a vaccine candidate. Another is take one particular protein. So there is another vaccine called Covishield or Moderna or Pharma. These vaccines, they were based on the antigen, where they have taken the surface protein of the coronavirus and they have given to the body as a vaccine candidate. There is another advancement that people are talking about, the epitope-based vaccine or peptide-based vaccines. Number of vaccines have come, particularly from Russia, which are based on the epitope-based. Our group is mainly focused on the epitope-based vaccine and antigen-based vaccines actually. So we have developed a number of tools over the years, and this is the concept that why we have the disease. Disease is caused by the pathogens. And then we have the 2 types of defense, non-specific defense and specific defense. In case of non-specific defense, it provides the first line of defense as well as it activates the function. Where the specific defense provides the B cell, memory B cells and memory T cells. So we have developed the tools for whole immune system rather than for a particular part. So here is the examples where we have developed the tools for the designing the vaccine adjuvants. Okay, this is very important that most of the drugs and vaccines are given with the adjuvants, because adjuvants activate your system actually. So these are the just example: peptide-based vaccine adjuvants, RNA-based vaccine adjuvants, and DNA-based adjuvants. These are the some of the examples. And these are the some of the tools. Let's say, for example, these are the tools for activating T cells or generating memory T cells. These are the tools designed for the B cell epitopes, okay, or memory B cells, and these are the tools for designing the vaccine adjuvants actually. Now, I have talked the one application of the peptide and protein in designing the vaccine. There is another example that you can use the peptide or protein as a inhibitors. Let's say, for example, our tool NTCP, initially it was first version was called NTCP, anti-cancer peptide, and recent version is NTCP2. Okay, we developed the method which can predict the anti-cancer peptides. These peptides can directly inhibit the cancer cells actually. Okay. Similarly, we have the tools like anti-hypertensive peptides. These are some examples. And what is the advantage? If you go to our website, we have the all how you design the different type of anti-hypertensive peptides. It will help you. Okay. Similarly, there is an anti-tuberculosis peptide. These are some of the examples where the peptide and protein is used as a inhibitors actually. Similarly, we can go for drug delivery. One of the peptide which is very useful is cell-penetrating peptide. These peptides have the property that they cross the membrane of the cell and deliver inside the cytoplasm. So if any drug is not going inside the cell, we can attach with the cell-penetrating peptide and it will deliver within the cell actually. So these peptides are used. So this software, Cell PPD, will help you to screen the cell-penetrating peptide as well as to design the new cell-penetrating peptides. And they are very user-friendly web servers. You can just put the sequence in the FASTA format. Rest of the things, the server will do the job. Okay. Similarly, we have the tumor homing peptides. These are the peptides, very specific peptides, which specifically bind with the tumor. They are not binding with other cell, they are binding with tumor. So if you attach the drug, they can deliver on the tumor side. And this software will help you to design the tumor homing peptide. Now, like any chemical, we have the ADMET properties, absorption, distribution, metabolism, and excretion. Same way, in the peptide also, there is a one word called toxicity. So we initially designed the software version 1 toxin pred. And now we have the toxin pred 2 or 3, where we predict whether a given peptide and protein will be toxic or non-toxic. Because it is important, if you have the toxic peptide, then it will not go into after the clinical trials, it will fail. So it's better to check their toxicity. But this will check only cytotoxicity, because there is a different type of toxicity. It will only check the cytotoxicity. Okay. Then next is hemolytic peptides. This will check the RBC. There are some peptide which lyse the RBC, which kill the RBC cells. Okay. Then these are called the hemolytic peptides. So this software, HemoPI, is designed for predicting the hemolytic peptides. So you can avoid these peptides in using as a therapeutics. Same way, some peptide can be, even in the foods, they can be allergic. Some protein may be allergic. So how to identify the allergens, particularly in the genetically modified crops, genetically modified foods? It's very important to identify the allergicity of a new protein actually. So this software, LGPRED, is designed for predicting the allergicity of peptide. Same way, this one is designed for diagnostics of the cancer, because in the case of the cancer, certain peptides are released in case of the cancer patient, but not in the normal. So this database maintains the information. This is a just example that we have many tools for peptide-based therapeutics. I cannot cover everything. We have more than 300 web servers and databases, but I am giving you some examples. If you are interested, you can use them. Let's say this B3PRED will allow you to design the blood-brain barrier peptides. Okay. So these are different. So I'm not going into the detail of them. Okay. My website is there. And thank you very much. for your attention.
Great, thanks so much. Nir Bental is on the call. Nir, would you like to take over the Q&A section?
No questions?
Yeah, sure.
Great,
Sure,
good
thanks.
to see you.
I'm sorry that I missed the beginning here, and the first talk. Are there questions?
So we have questions in the Q&A if you can see it.
Okay, the question is, do you have the proof against the designing the peptide? Okay.
Yeah.
So here we have to understand that we have used only experimentally validated data sets. And one part of the data set, which is not used for training or testing, we have used for the independent validation. This is as good as experimental validation.
So can I elaborate on this? So these unused data, how different are they from the data that you trained on?
Okay, so what we do normally, we take the whole data and we sort on certain criteria. And then we take the first in independent, then next 4 or 5 in the training, then next in the, so the data is well distributed. All type of data is there in the independent data set. The distribution is similar.
Okay, thank you.
Can I ask a follow-up question?
Please do, yes.
Excellent, thanks. That was a great talk. So I was really curious on, oftentimes we have an example of a protein where we know what it binds to, but we may want to identify a peptide that maybe binds better. And so is that a possibility using your databases to identify similar peptides based on a target that you already have? And if so, is there also a way to be able to predict affinities that would be really helpful in looking at things like inhibitors?
Okay, so far we have no such type of method. Maybe in future we will think.
Are there more questions? Okay, if not, I will just and again I'm sorry for being late. I'll just introduce myself briefly, saying that I'm computational structural biology from Tel Aviv University, and I'm here as within my role as senior editor with Protein Science. I want to share a slide that you saw before, just to clarify, maybe, and to make sure that the message is passed. Do you see the slide? I hope so.
Yes,
so,
we do.
Yes.
Yeah. So these are tools papers. This is and you can see that there is a constant increase in the amount of tools paper that were published. For the most part, these are computational tools, but there are some experimental protocols and such. The link here is for the last one that was released, that Tools 25. And this is what we have for Tools 26. We have a total of 18 papers that were accepted, and 9 are in review, and 36 different people promised, or research group promised us to send papers, and in addition there are some maybes, but we aim to reach 100 if possible. So if any of you who are in this webinar or who are going to watch it, if you have interesting suggestions for what could be a tool, please approach. We can consider, because we want to accommodate more papers. The idea is to look for papers that with tools, either computational or experimental, that are going to be very helpful to the community. Okay, and with that, I'll stop my share and then shift to the next speaker. So is it Matteo?
Hi.
Okay, so please, Matteo.
Yes. 1 second. It should be working, right?
Yes.
Hi, everyone. Good morning, good afternoon, good evening, wherever you are. I'm Matteo Tiberti from the Cancer Structural Biology Lab, and I'm going to talk to you about BVSP, our computational framework for the protein invariant effect prediction and interpretation. And first of all, I would like to thank you very much, Protein Science and the Protein Society, for inviting us to give this contribution, which I hope we stir quite a bit of discussion. So a few words about me, about us. So I'm a staff scientist at the Cancer Structural Biology Systems Biology Lab. This is a laboratory headed by Erna Papaleo in Copenhagen, in Denmark. It has two names because we are a lab with two homes. We are partially at the Danish Cancer Society, which is a charity which fights against cancer, and at the Danish Technical University. In general, our aim is to cover comprehensive research cancer pipeline from cancer patient data to identify cancer driver genes, and then in-depth investigation of these targets and the effect of their mutations. And to do this, we use fundamental methodologies such as bioinformatics, and of course, structural biology and biochemical and physical assays. So I'm just get to a laser pointer. Yeah, but today we're going to in particular focus on this part here, which is the investigation of functional mechanisms and effects of mutations. This is because, of course, mutations are the prime cause of many diseases, including cancer. And now, because of genome sequencing has become so cheap, and because gene panels being highly available, we have so many variants that are known to be found in patients. However, we have much less knowledge about what these variants actually do. So if you look at this plot here, this is ClinVar, for instance, and we see that even today, the majority of the variants in ClinVar are classified as VOS, so variants of uncertain significance. So basically, we don't have enough evidence to know what these variants do. And not only that, but this number has been increasing on and on in the years. And so the interpretation of the variants is lagging behind. Of course, interpreting variants will be very useful because of different reasons, to design therapeutic strategies, to stratify patients, and identify variants connected to drug resistance and others. Of course, there are many, at this point, computational methods that can predict the effect of mutations. They also work pretty well in general terms, but they don't give us a mechanistic explanation of what the variant does. So we usually get how likely is a variant to be pathogenic or to be damaging response, but not really why or what does this variant do? What's the intimate mechanism that this variant damages? And that's because, of course, there are many ways that the mutation can alter a protein. It can affect changes in structural stability, leading to misfolding, leading to degradation, so less availability in the cell. It can disrupt interaction with other proteins and biomolecules, just for disrupting pathways or regulation. It can interfere with regulatory mechanisms such as phosphorylation modifications. Of course, function of protein can be altered, so enzyme activity can be altered, for instance, including loss or constitutive of activity of constitutive activation and long-range effects that work through allosteric mechanisms. So based on this, we set out to try and answer this question. Since there are so many aspects that a mutation, a variant can affect in a protein, can we design a so-called structural framework, a framework based on structural biology to systematically assess how each mutation impacts each of these protein properties? And in order to do that, what we did was we collected a number of methods and methodologies, each of them designed to look at the different aspect of the effect of a mutation.
we collected these methods that predict, in general, its potential pathogenicity. You can see a little list here. But more in detail, we wanted to use tools to quantify different functional or structural effects of mutations, including effects on stability, so folding and unfolding, on binding, on macromolecules, on other macromolecules, so other proteins or DNA, for instance, effect on velocity, changes in protein dynamics and conformation changes. And we would also like to include in the mix some data from experiments. And all of these, of course, since we're talking about many mutations in a protein, would need to be done high throughput. So we need high throughput strategies to handle such a large scale calculations on many, many variants on each protein, which leads us to the desire to have standardized workflows. This philosophy led to the creation of MAVISP, which is our multi-layered assessment of variants by structure for proteins, so-called computational framework. So MAVISP is a collection of methodologies or protocols, each of which is designed to tackle to predict the effect of mutation for a different aspect. Okay, you can see the figure is divided in two. We'll see first here simple mode, and I will tell you the difference. Each of these frameworks, so-called, is completely modular. That means that each aspect can be predicted separately, and different modules do different things, of course. So, for instance, we have some modules that are designed to identify the most suitable protein structures to then use the other modules, or in the interactive modules, for instance, that identifies interactors for our protein. We have some other modules that plug in variant effect predictors and experimental data. And then based on this, we have some modules that predict the effect of mutations on stability, whether they are part of the early folding portion of the protein, predict effect on PTMs, on interactions with other proteins, on long-range effects, functional sites, and so on. So all different aspects that are predicted independently. And this can be done starting from two starting points. In simple mode, we do so by starting from a single protein structure. So this is our high throughput, more simple mode, as it's called. While in ensemble mode, we start from an ensemble of structures. And that's because this allows us to incorporate also dynamical information in our calculations. And for ensemble mode, we usually use molecular dynamic simulations as our input. So how, given this structure that I just showed you, how do we get from the starting point to our predictions? So this is the MAVISP processing pipeline. You can see here that what we do is that we select a certain protein, and then we use different tools, as I said, to predict different aspects of our mutations, which are part of the MAVISP toolkit. These allow us to get to a certain data set that we see here, which contains the output of each module. And this is done semi-automatically. So we do most predictions automatically in a pipeline built with SnakeMate, but there are also some more manual aspects. Once we have all the data, we have a Python package that takes all this data, compiles a database fundamentally. So we get a database which contains, for each mutation, all the output of all the modules, including both the values eventually that this module have given us, and a potential classification. And from here, we get to our output that can be seen by our users, let's say. So we have a website where we upload all our predictions are available. I will show you that later. We have a downstream analysis modules, which perform some analysis that show us then the final results and the compilation of the report. MAVISP has three types of people that work in it. One is the biocurators, which are the people that perform all the analysis here in the first pass of the pipeline. One is data managers that take care of doing the last part, of importing this data and then making it available, and developers who develop the tools that are part of MAVISP, which can mean either here, so for the tools that do the predictions, or here, so for the tools that digest all the data and then produce the final result. Currently, we have a MVSP website which contains all of our predictions that we've done so far, and we keep working on it and adding new proteins. So far, we have about 1,200 proteins with more than 2 million variants, so quite a number. This is just a slide to show you how this would not be possible without a specific tool set. Over the years, we have developed many different tools that do all these analysis. Some of them automatize the different analysis that we want to use, and some of them are designed to use to implement specific methods and do specific analysis by themselves. This is just in case you're curious to see some of them. And also, we at the lab, as I was anticipating, we also have a small wet lab. So we also do some cellular in vitro assays for protein-protein interactions, especially in terms of protein-protein interactions. So to see the effect of mutations on the interaction between proteins. And then we are also moving our first steps towards implementing multiplex assays for protein pointers that will come. But we also take data from available databases such as MaveDB or ProgDream, which contain data from multiplex assays for different functional readouts, and of course, literature mining. Here, I wanted to show you a little example of what we get in the end of the whole process. So this is the P10, which is a tumor suppressor protein. And this is a case in which we are looking at variants that were available in ClinVar. So if you see here, this bar here is all the variants that were available for this protein in ClinVar. Well, some of the variants that were available in this protein in ClinVar. And the ones in red were classified as damaging, and the ones in green, were classified as pathogenic, and the ones in green were classified as neutral. And you can see here on each row is the output of a different NavSP module. So for each mutation, we see the output of each module fundamentally. And the different color tells us whether the module predict this mutation to be damaging, to be neutral, uncertain, and so on. In this case, for instance, we can see that in this case, this protein was predicted as pathogenic. We can see that our variant effect predictors in green also agree with this assessment. But we can see also that our more functional, if you want, predictions also, and also our stability more based predictions all agree on this variant being neutral, so not being damaging. And here we have another case in which is the exact opposite, and we can see that all our predictors do agree that this variant should be damaging. But we can also see that there are a couple here, what we call mechanistic indicators that tells us that this variant is damaging. So now we know not only that this variant is likely to be damaging, but we also know why. So for instance, this variant is likely to be stabilizing these protein structures and also have some long-range effects. And all of this data can be compacted a little bit more into this second representation, which we just see the mutations that are found to be damaging and a more simple classification. So these are two most common types of output that you will see from RSP. And this is exactly the same, but in this case, we didn't use variants from ClinVar, but we did the same analysis on variants of uncertain prediction, of uncertain significance. And so we see that in this case, for instance, these two variants are predicted by some of our variant effect predictors to be damaging. And we have a mechanistic explanation of the effect. So in this case, this would be damaging for stability. Here, I just wanted to share a recent paper that we published. This is actually the work of a PhD student of our lab who is called Matteo, and this work has been recently accepted in molecular oncology. And this work is, in this work, we have applied MVSP to poly-ApoLD1, so which are two important DNA polymerases, epsilon and delta. They play an important role in DNA replication and ensure the replication fidelity through their proofreading activity. And of course, protein variants in poly and POLD1 could affect their structural integrity and function. We assessed a total of 43,000 and something variants for poly and more than 21,000 variants for POLD1, so quite a bit. And we use for this both simple and ensemble mode. You can see here on the left, more or less a scheme of what we did. So we started from a set of variants that came from either CleanVar, which were found to be pathogenic or benign, so classified in CleanVar, and those were just analyzed using the MSP mechanistic indicators. But for other variants, such as those that had a loose or conflicting classification in CleanVar or others, were first filtered using our web. So we only considered the variants that were classified as damaging for our webs, and then those were then considered for the mechanistic indicators. And this is just, I can change slide, okay. And this is just an example which is similar to the ones we saw before. In this case, these are all pathogenic CleanVar variants. In this case, MSP and SAML mode identified 3 variants. You can see here in the squares that were classified as damaging, and also at a mechanistic indicator. So you can see that this one, for instance, would be a variant, a residue involved in early folding. This would be a variant that affects binding with DNA, and this would be a variant that affects stability, for instance. For other variants were identified as having a certain effect for stability, and 2 of them seem to have more conformation-dependent effects, so we added more of a deep dive using molecular dynamics to really understand what is going on. And we found out that by comparing molecular dynamic simulations of wild-type mutant protein, those variants were affecting the dynamics and also the structure, partially the structure of the active site. So we think they're likely to be affected there. And this, if I can turn the page, yeah, this is a similar story, but done on VUS instead of the CleanVar variants. So after filtering, we found several variants of interest, which had a mechanistic explanation. And then we focused on variants that were particularly damaging for stability or binding and close to functional sites. And you can see here in these plots, for instance, that the one in the red box, they are damaging for binding to DNA. So they're found to be damaging and to be likely to disrupt the binding with DNA. While the ones in orange were found to be disrupting for the binding of other proteins, other cofactors that usually are important for the accuracy of DNA replication. Something else we do with MAVISP is that we use MAVISP to compare ourselves with experimental data. In fact, MAVISP also includes a module to import experimental data in the framework. And this data can be used in 2 ways. They can be just used as additional layer of information, or they can be used for benchmarking our methods. Here, I've shown you a simple example of using the stability module on 10. And here we compare the performance against experimental multiplex assays that measure cellular protein abundance, versus our stability module. And we could find that in our case, we compare it also with gem and mask, so to VEPs. We can find that we have a likely better performance than them. And then I wanted to, if I have time, what am I 16 minutes, I can show you something. I just wanted to quickly show you a little bit of my MAVISP website. I can show you live, actually. Just a second, I need to change the screen. So this is how you access the MAVISP website. You can find the URL for it on our paper. This is the MAVISP website itself. This is the welcome page, and then it's the most important section is this part here. You can see simple mode and ensemble mode. So we differentiate between the 2 modes. So the results are independent from one another. Here you can see a list of protein. Of course, you can filter up here, listen for 10. And if we do so, we can have access to the full data set. So you can see here, I will switch it to full data set view. And you can see here that this is basically a table that has mutations as the key, if you want. And then all the properties and all the modules that MavisP all the MavisP modules that have been performed on this protein are available here, and all the results are available either in numerical form or as a classification and so on. And that's a simple way to access our data if you want to look at it. But of course, we can also produce our own classification, which are dot plots that you've seen earlier. So for instance, here I can select a few mutations. Oh. And you can see that this shows exactly the plots that you were looking earlier. So these can be very useful if you have some specific mutations and you want to look into them and see what MavisP thinks about them. Yeah, there are another couple of representations, but I think I'm a bit late, so I don't think I'll be able to show them to you. But they are here and here if you want to try them yourself. This one in particular, which you haven't seen before, is designed to visualize to identify on the protein structure where hotspots for a certain type of effect are. So, for instance, to identify which residues are hotspots for stability or hotspots for long range, for mutations that affect long range effects and so on. Let's stop sharing. I'll go back to the slides. Oop, I go back. Okay. So we're nearing the end. A couple of concluding remarks of what we've seen. So we have seen how to use MavisP to understand different mechanistic aspects of our effects in proteins at scale, so at high throughput, or as high throughput as we can. MavisP can be then used to prioritize variants for experimental studies, and also using the mechanistic indicators to identify what the most suitable biological readouts for your experiment, for an experiment validating the predictions will be. We also have, as just shown you, a large database which is freely available for everyone. So it's important for us to create and maintain a data set of features from protein structures. This can be then leveraged on, for instance, using machine learning models or validation using other methods. And of course, our tool, as I told you, is modular. So that means that modules can be taken in and out, and we would be very happy to be able to expand and improve over time our tools so that more mechanistic indicators can be added, or some tools maybe can be swapped out for others if they perform better or are faster. A few things about our work in progress. So we're trying to assess variance effects on protein-drug interactions. We would really like to have a machine learning approach that will confidently predict binding free energies. Of course, that's a big ask at the moment. We would like to include, as I was saying, additional methods and sampling approaches. We are organizing training activities for bike creators from external groups, talking of which we are organizing a workshop for 2026. This will be probably early September. And this will be for curators, so for people who want to become part of the MavisP group and want to create their own proteins, for instance, using MavisP. And if you're curious about that, there is a page on the website, or you can just write me or write us. And of course, we will also try more and more to reuse available resources, for instance, mining or using in vitro trajectories for the analysis. I also wanted to say that we have a pathway for inclusion of new approaches. So that means that we have a policy in place that allow us to have a systematic pipeline to add new approaches and new models to MavisP. And of course, we would be very, very happy to hear from you, and in case you have methods that you think will be useful to the framework. And that's it. That's what I wanted to say. And I thank once again the Protein Society for inviting us. And these are the people that have participated in the project, probably not even all of them, but it's a good start. Thank you.
Thank you very much. I look for questions. I don't see any for now. So let me ask a question of my own. So how well do you do in differentiating between gain of function versus loss of function mutations?
Yeah, that's a very interesting question. Yeah, so currently our approach is much more suited for loss of function mutations. That partially because some of our tools are more suitable for loss of function mutations. For instance, all the tools that predict changes in stability are more suitable for that. And also because, we in general focus first on, for instance, on proteins that are pathogenic when mutated in general. So that they lose their function when mutated. So I would say we are more geared towards that, but we definitely would like also to see the other aspect more.
Okay. And then another question, I'm sorry, I would have given chance to others, but I don't see anything written. What about, can you elaborate some about your ideas on adding interpretability on like mechanistic understanding of the mutations? What do you have in mind in this?
Well, I mean, the idea is just that we would like to add as many tools as possible, each of them adding a new layer. So each of them would be responsible for their own type of mechanism, let's say.
I see.
So that's more or less the idea. So we add layers by layers, trying to cover as many aspects as possible.
I see.
So that would be our mechanistic interpretation, if you want.
Okay. And are there other questions?
One maybe.
Oh, I see here. By Jonathan.
Yeah.
So you said you're interested in improved free energy perturbation predictions. What technique do you currently use for effects on ligand binding?
So we have a project which is about trying to identify resistance mutations. So in terms of binding. So that means trying to predict how the free energy of binding changes when mutating your protein. So free energy of binding to a small molecule. And to use that, we are trying an approach similar to thermodynamic integration. Of course, we're happy to see other ideas.
Okay, are there more questions? Okay, so thank you very much, Matteo.
Thank you.
And now let's move to Luca. So welcome, Luca Genz.
Hi.
So Luca is our last speaker for today.
Can you see my screen on my slides?
Yeah.
Perfect. Thank you. So hello, everyone. Also from my side, I quickly introduce myself. My name is Luca Genz, and I'm a final year PhD student in the group of Professor Maya Topf at the Leibniz Institute of Virology in Hamburg, Germany. And I would like to start with thanking the organizers for giving me the opportunity to present my research today. My talk is titled Assessing Scoring Metrics for AlphaFold 2 and AlphaFold 3 Protein Complex Predictions, which is also the paper that was published in the Protein Science Tools special issue in 2025. I would like to start with a very common scenario, especially for structural biologists, which is that we have two proteins in a cell, and we don't know the structure of these proteins, and more importantly, we don't know the structure of the complex that is formed by these two proteins. And resolving the structure experimentally can be very time-consuming and also quite expensive. So what do we do? And there is this tool that was already mentioned a couple of times today, which is AlphaFold or AlphaFold 2 and its successor AlphaFold 3, which was even awarded the Nobel Prize in 2024, specifically for the prediction of monomeric proteins on a near experimental resolution level. And just yesterday, it was announced that the AlphaFold database will from now on also include protein complexes, or more precisely, homomeric protein dimers, which makes the question that we are trying to answer even more important, and that is, if we have a protein complex prediction, how can we tell that that is our real structure? And to answer this question, we need a score. And this score has to measure the quality of the predicted complex. And also what we are doing is assessing the three prediction methods, or three different prediction methods. That is collab-fold with templates, collab-fold being an implementation of AlphaFold 2. And with templates means that it has access to the protein data bank in the prediction process. Then we also wanted to assess collab-fold without templates, so AlphaFold 2 without access to the PDB, and then also AlphaFold 3 with access to the PDB. And to actually find a score that reliably measures the quality of the predicted complex, that is basically the aim of our research, and that for all the data sets that or just the prediction methods that I just mentioned, which are CFT, CFF, and AlphaFold 3. To assess the performance of the score and also the performance of the prediction methods, we need a data set. And the data set that we tested on is what I call the benchmark set. And we started with filtering the PDB for heterodimeric complexes that were supposed to be high resolution. And very importantly, we also filtered for a release date that was after the training cutoff date of AlphaFold 3. And we ended up with a total of 684 complexes. In the next step, we also wanted to make sure that these complexes are really interacting based on distance. And for that, we used PigCluster. And PigCluster is a UCFS Chimera X plugin that we developed that also exists as a command line tool. And this identifies protein interfaces based on distance. And it also provides a range of scoring metrics for the analysis of the detected interface, which I will talk a bit more into detail in a few seconds. And then also PigCluster works not only for protein complex structures, but also for models generated by AlphaFold 2 and AlphaFold 3. In the next step, we filtered out antibodies, and we ended up with 343 complex structures. And in the next step, we also wanted to reduce sequence redundancy in our data set. For that, we used the tool CD-HIT and ended up with 268 complexes. And then we started predicting the target complexes using collab-fold and AlphaFold 3. But we had something that I call the AUBA problem, or the asymmetric unit biological assembly problem, because for some of the cases in our data set, we saw that the asymmetric unit deposited in the protein data bank differs from the biological assembly. And when we do our prediction, we don't really know if the prediction is going to resemble more closely the asymmetric unit, which we compare to, or the biological assembly. And this created artificially low scores when the prediction resembled more closely the biological assembly. And for that reason, we decided to filter out protein complexes for which the asymmetric unit differed from the biological assembly, and ended up with 262 complexes, which were then split into 223 complexes in our benchmark set, and we also had a test set. Besides the data set, we also need a ground truth to assess our scores, and that means that we need to compare our template to our prediction in a couple of different ways. One way being to look at the interface specifically, and there is this really nice score that superimposes the template and the prediction at the interface that is called IRMS. And we also needed to look at the contexts that differ between templates and the template and the prediction in the interface. And there is the score called FNAT that does that for us. Also, we wanted to look at the difference in global fold and orientation. And for that, there's the score LRMS. And to our advantage, there is this score that is called DocQ that combines all of the three scores that I just talked about. And what is even more nice is that for DocQ, there are classification cutoffs based on Capri. And these classification cutoffs allow us to classify the predictions into 4 categories, namely incorrect, acceptable, medium, and high quality. And what I also want to mention is that we use this cutoff of 0.23 to differentiate between the incorrect fraction of our predictions and summed up acceptable, medium, and high quality predictions to what we call the correct predictions. So with that, we had a ground truth and our benchmark set, and I would like to start with having a look at how well the different prediction methods performed on our benchmark set. We assessed that with the DocQ classifications. And from this plot, you can already see that the fraction of high quality predictions is the highest for AlphaFold 3, and also the incorrect fraction is lowest for AlphaFold 3, which indicates that AlphaFold 3 actually gives us the most reliable predictions. We also had a closer look at the incorrect fraction only, because if we predict our target structure, we get 5 models per run, and one fact that we wanted to investigate was how consistent the prediction method basically is in the quality of the prediction. And here you can see that AlphaFold 3 actually has the highest fraction of models for which all 5 models are classified as incorrect, which indicates that AlphaFold 3 predictions are the most consistent ones in regard to whether a model is correct or incorrect. Then we also looked at the consensus of the 3 different prediction methods, and you can see that for the incorrect fraction, we have a consensus of roughly 50% between the 3 prediction methods. And the lowest fraction of incorrect models is for AlphaFold 3, where only AlphaFold 3 predicted an incorrect model. And for the correct fraction, we see a very high consensus of nearly 80% between the 3 prediction methods. And we see the highest fraction where only one prediction method was capable of predicting a correct model for AlphaFold 3. These results align with what we just saw, that AlphaFold 3 is the most reliable prediction method. However, if we split the correct fraction again to acceptable, medium, high quality, and look precisely into the differences between the incorrect and the acceptable fraction, we can clearly see that the consensus for the acceptable fraction is much, much lower, going down to 14%, where the 3 prediction methods align, which indicates that near the incorrect threshold, it really makes sense to compare the outcome of all 3 prediction methods, because this may yield the most reliable result. We also had a look at which factors are driving the prediction success and also the mispredictions. And we first of all looked at the B factor in the incorrect and the high quality fraction of predictions. And one can clearly see that the mean average B factor for the incorrect models is higher compared to the high quality predictions. We also looked at the interface size, and you can see that in here the maximum of the distribution for the high quality models is towards a larger interface size compared to the incorrect fraction. This indicates that an interface of lower flexibility and larger interface size may yield more reliable predictions. Then coming to the scores that I talked about, we wanted to investigate 7 commonly used assessment scores. The first 2 are IPLDDT and IPAE, IPLDDT being the interface-specific version of PLDDT, which is a confidence measure that comes from AlphaFold, and IPAE being the interface-specific version of the PAE, which is an error measurement that comes from AlphaFold. And we computed these 2 using our already mentioned plugin PigCluster. Then we also looked at PTM, IPTM, and model confidence, which are also confidence measures coming from AlphaFold directly. IPTM being the interface-specific version of the PTM, and model confidence being a combination of these 2. And then also we looked at PDoQ2 and VORAI-FGNN. VORAI-FGNN I will from now on just call VORAI-F. And these are external quality predictors. So then we assessed the performance of these scores using our ground truth measure DocQ, and we calculated the Pearson correlation coefficient for all individual scores with the DocQ across all of the 3 data sets. And first of all, I would like to mention that the IPTM and the model confidence showed the highest Pearson correlation with DocQ across all 3 data sets, meaning that most probably IPTM and model confidence are our most reliable assessment scores. Also, to our surprise actually, because we just saw that AlphaFold 3 gives us the most reliable predictions, but coming to the scores, we get the highest Pearson correlations for running CollabFold without templates, which means that most probably the scores run best for CollabFold without templates. And then lastly, I want to mention that the interface-specific IPLDDT and IPAE outperform their global counterparts PLDDT and PAE, which was not really a surprise as we calculated the PCC with DocQ, and DocQ mainly looks at the interface. And now we have the scores and we know which scores to use, but scores are just numbers. But most of the time we want something that tells us, do I continue with this model or do I try again? And for this reason, we wanted some cutoffs that are capable to distinguish between correct and incorrect models. And we determined cutoffs using our ROC curve analysis. And I want to show you just one example of how we did it. So you plot the ROC curve, and then for IPLDT, for example, the point of maximum distance from the diagonal gives us the best ratio between false positives and true positives, and therefore is the optimal cutoff for this score. And we did that for all of the scores and all of the across all 3 prediction methods. That gives us this nice table that allows us, based on the score, to distinguish between correct and incorrect models. And I just mentioned that IPTM is among the 2 best performing scores. And actually for IPTM, there is a gray zone known by AlphaFold, which is between 0.6 and 0.8, where AlphaFold claims that it can't clearly be said if a model is good or bad. And now with our cutoffs, we can actually say if a model is correct or incorrect. And I brought a few examples to showcase that. And in this first example, coming from the CollabFold with template dataset, we can clearly see that our IPTM value is below our determined cutoff, aligning with the determined DocQ, meaning that this interface is actually incorrectly predicted. Then this is one example from running CollabFold without templates, where we see that the IPTM is above our determined cutoff, aligning with DocQ, which means that the interface is correctly predicted in this case. And this is another example from the AFOD3 dataset, where we see the IPTM is in the gray zone, but below our determined cutoff. And again, aligning with DocQ, saying that this is an incorrectly predicted interface. So I'm already coming to the conclusion. We curated a benchmark set consisting of 223 protein complex structures and the corresponding models. We benchmarked the accuracy of the prediction methods, CollabFold with templates, CollabFold without templates, and AlphaFold3. And we figured out that AlphaFold3 gives us the most reliable predictions for our benchmark set. Then we assessed the performance of our 7 commonly wide assessment scores, and we found that IPTM and the model confidence seem to be the most reliable assessment scores. And something else I didn't have time today to talk about is that we did create a combined scoring approach that we call C2Q score, that combines 5 of the individual scores, and by that trying to create a more robust approach in assessing protein complex predictions. And if someone wants to try it out, C2Q score, but also all individual scores were integrated into PigCluster, and PigCluster can be easily downloaded from the CameraX toolshed. Then also just a quick side note, I want to mention our newly developed plugin ProNA3D, which extends the functionalities of PigCluster not only to protein complexes, but also to protein nucleic acid and nucleic acid only complexes. And it also has a couple more features like topology plotting for nucleic acid structures or 2D interface graphs. So last but not least, I would like to thank the entire TOPF group, and especially Maya, and also my second supervisor, Kai Grünewald, and of course you for your attention.
Thank you very much, Luca. Are there questions? So just to make sure I got you right, so the main problem in predicting protein interfaces is that in sampling the correct interface, right? I mean, once it's there, then with your definitely with your revised score, you'll be able to you'll most likely be able to detect the correct interface, correct?
I hope so, at least. I mean, it definitely depends on what's the outcome of AlphaFold. But before having the cutoffs, I saw a lot of papers reporting the scores, but they couldn't really say if this is a good or bad score. And with especially providing cutoffs, we tried to make it easier for people to decide if an interface is correct or incorrect.
Mm-hmm. And in cases where you based on your score, you figure out that the interface is incorrect, can you somehow suggest where the interface is? Can you in that case maybe suggest a new method to model it and then test that other interface?
I mean, detecting the interface in the model itself can be done with PigCluster. But I guess then more the problem is the prediction method itself. And right. As we've seen, we saw that in some cases, actually an older implementation like AlphaFold 2 gives us better results. So especially in these cases, my suggestion is always to try different methods and see the consensus and also where there are differences maybe. And then, yeah, hope for the best that the prediction gets better.
Okay, so I see a written question by Alejandro. Have you analyzed how the scores perform for proteins that have low number of homologs? less than 100 sequence with less than 95% identity, for example?
So, oh, I can't see the question anymore. I mean, we did specifically filter for, if I get the question correctly, I can't see it anymore, unfortunately. Ah, there it is. No, we didn't. I mean, we did filter for sequence redundancy in general, but we didn't look specifically at the MSA that's coming from AlphaFold. No, we don't look into that. But it's definitely something that would be interesting because just from guessing, it's more harder to predict, of course, these proteins.
Okay. Are there more questions? Maybe Matteo or Gajendra or Heather, you seem
Yes,
online now.
I was going to say
Maybe,
I haven't. Sorry, Matteo. Do you want to go first?
no, go ahead, go ahead.
Okay. So, in relation to looking at interfaces, I have an interesting problem that maybe is solvable by using your platform. We have a novel structure of a dimerization domain, where we can see the interface within the dimerization domain. But of course, like it hasn't been put in the PDB because we're still kind of working on it. But we know that this family of proteins have very few homologs, and we also know that they heterodimerize. And so, is it possible to predict heterodimerization just in general if you have a sense of two proteins that homodimerize, but then also can heterodimerize, especially when there's limited sequence similarity between the two dimerization domains?
So you mean if you can predict with which other proteins this domain would form a complex?
Exactly.
So I mean, if there's data on oh, I'm sorry, I think I'm lagging again. I'm just switching my video off. I think in general, you can always try to predict the complex for if you have any guesses, if there's any experimental data known, like yeast to hybrid or something like that, and then see if you get good scores. But what's really important to mention coming to my analysis is that we look at correct, if the model is correct or incorrect. We can't really see from these cutoffs if the complex actually exists. That's a very different problem that a lot of people are working on because that's much harder to say actually just from the numbers. Because it could be also just due to homologous structures, right?
Yeah, thanks.
More questions? Matteo?
Yeah, just have maybe a comment. I was just thinking about Mavisp, and I think your work will be very valuable, well, to the community in general. And I think also for us, because of course, there are cases in which we want to, as one of the Mavisp modules is a module that evaluates how mutations affect binding between proteins, of course. In that case, you need to start from the protein complex. And of course, there are not many, I mean, there are not as many as we would like of those around, right?
Yeah.
So having a good way to assess whether your model is good would really, a robust way to assess whether your model is good is very valuable to me. We'll definitely have a look. Thank you.
That's great. Thank you.
Are there more questions?
There's one in the Q&A.
Yeah, I can see that. So do you see it? Have
Yes,
you
I see
directly?
it.
Okay, yeah. So I'll read it and then you can answer it.
Okay.
Have you directly compared AlphaFold 3 predictions with docking-based methods such as ClassPro or Haddock under equivalent conditions? And what matrices would you use to evaluate performance? like RMS deviation or interface quality?
That's a great question. No, we haven't, because we wanted to mainly focus on the prediction methods and not on the docking algorithms. Also because it was easily comparable when it comes to scores. So the AlphaFold models gives us the same scores like PAE and PLDT, and especially PLDT and IPTM are measures that are often reported in papers when they show predictions. And in general, I would always go for the DocQ metric, because that's a metric that's been out there for very long already, and it has been used in a lot of papers. And you especially for DocQ, as I said, you have these different categories, which makes it really nice to assess the performance of the score. And it integrates basically, RMSD, but interface-specific RMSD, which is this, which I quickly mentioned, IRMS, that is part of the equation of DocQ.
Okay, thank you. Anything else? Any more questions? Okay, in that case, let me close the session. Thank you very much for everybody, all the great speakers, and for all the participants. And I look forward to when Tools 26 will come out. And as I say, if you have suggestions for what you think is a useful tool to be included in Tools 26, then please call me. Contact me.
Thank you, and thanks for the organization.
Thank you
Thanks,
very much.
everyone.
Thank
This
you
is
very much.
great.
Thanks a lot.
Thank you.
Bye.
Bye-bye.
Bye bye.